In the last chapter, we started talking about cells, and in that, we talked a little bit about membranes. We're actually going to spend a whole chapter talking about membranes because they're really important. They're what separates the inside of the cell from the outside of the cell. That means everything that comes and goes into and out of the cell has to pass by a membrane. And there are a lot of different ways of moving things and rules for what can move where and how and when. This also is really important because when we talk about things like microbes, viruses, bacteria, things like that, they might want to get inside of your cell. And how they do that, it's good to know how they do that because that might be a way we can stop them from actually getting inside. So if you're interested in disease and microbiology, membranes, you're going to have to know a lot about them as well. So we're going to start by talking about the components and the structures of the membrane, and then we will spend the next few sections talking about different ways of moving things across the membrane. So we'll talk about both passive and active transport, and then moving really large stuff. We call this bulk transport. So what does the cell membrane actually do? It, of course, defines the boundaries of the cell. Anything inside of it is inside the cell. Anything outside of it is outside of the cell. It also controls what can enter and what can exit. We talked a little bit about how membranes can help join cells together with those joints, tight junctions, things like that. Membranes are really critical in receiving external signals, so molecules that come along and tell the cell something, or things like nerve impulses and stuff like that. And also, they're very important in the immune system. Our body has a whole system of membrane proteins, basically, that are designed to recognize your own self compared with other things that are not yourself and deal with those non-self things. So, a very important structure here in the membrane. We'll start out by talking about the structure and its components, but I want to also give you some examples of why understanding membranes is important. So we'll talk about the fluid mosaic model, as we call it. We'll talk about phospholipids again, the proteins that are involved, and some carbohydrates, and then also talk about membrane fluidity. The membrane is not rigid. It can flow and move, and that's a really important feature of it. But let's start with an application here. So you may have heard of this virus. This virus is called HIV, human immunodeficiency virus. HIV, like all other viruses, needs a cell to replicate in. Viruses cannot replicate themselves. Now the problem is your cells generally don't want viruses in them, okay? It's not good for them. So HIV needs to come up with a way to get into your cell, and it does that through trickery. On the surface of certain cells, in the case of this example, white blood cells, we have a special receptor called CD4. Okay, this CD4 is found on white blood cells. HIV has some proteins on the outside of it that bind to CD4. Now, CD4's job is a receptor. HIV has evolved proteins that look like the signal CD4 is looking for. So they're mimicking that structure. And when they attach to it, they bind to the cell, and the cell thinks that it has come in contact with something it is supposed to let inside of it. It does not know it's been tricked, right? So HIV can then enter into the cell, and then it's game over. It will integrate with the genome, and then it is there forever. HIV also makes copies of itself inside of the cell, and then it uses some of the host cell membrane to package itself up, and then new particles float away and go infect other cells. So viruses exploiting cell surface receptors and the membrane transport is just one example of why understanding membranes is so critical. The membrane itself, as we said, is a pair of phospholipids on either side. We have the hydrophobic tails hiding away and the hydrophilic heads pointing out towards the water, either outside the cell or inside the cell. Embedded in this are tons of different proteins and carbohydrates, lipids, things like this. The membrane here is actually fluid. So we call this a fluid mosaic. It has stuff in it. These things don't necessarily stay put. They can flow throughout this lipid membrane here. There are a couple of different types of proteins that are involved here. There are integral proteins, which actually go across both layers of the lipid bilayer, like this protein channel here. Those are integral proteins. And then there are peripheral proteins. Those are attached only to one surface. In this case, we have like this glycolipid that's attached just to the outer surface here. So this fluid mosaic, it's constantly moving, and these molecules are actually moving and flowing around one another. They're not rigidly attached to one another. It's purely that fear of water that's holding this all together. There's some other things like cholesterol and carbohydrates that are embedded in here as well. We'll talk a little bit more about cholesterol in a moment. Okay, we need to review our phospholipid structure because it's really important to the structure of the membrane. The phospholipid, it has the glycerol here. One of the carbons is attached to a phosphate group. That makes this whole region here hydrophilic, water-loving. And the tails, the two fatty acids, are hydrophobic here. You can also call these two regions polar. The hydrophilic head is polar, and the hydrophobic tail is nonpolar. This makes phospholipids amphipathic. They're both hydrophilic and hydrophobic. So when we get a bunch of them together and we throw them in water, they will self-organize into a structure. Here you can see a lipid bilayer that forms a little spherical structure if you get enough of them together. That creates an inner space here. I think you can see where this is going. This is like the beginnings of a cell, right? Technically, if you have just enough, they could form a little single layer sphere, but that's not going to be relevant in biology, right? Because we need this double layer here because we need water inside of the cell. So the lipid bilayer is the form we find in cell membranes. This is occurring in what we call an aqueous environment. I throw that term out there because you will see that a lot in science. Aqueous means containing water. So outside the cell is aqueous solution, and inside the cell is aqueous solution because it contains water. There are no bonds holding these layers together, so they are free to flow around one another. They only very rarely flip sides, but they do flow left to right, back to front quite frequently, but very rarely do they flip-flop on sides. All right, let's look at a little animation of this occurring.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, so I think that animation really shows the fluid nature of the membrane, and there are all kinds of things moving around in there. They're not static. Some of the things embedded in the membrane are things like integral membrane proteins. We have different things, and they have to go across the membrane. These proteins, because they're going across the membrane, need to have certain properties that will not, interfere with the membrane or will gel well with the membrane, essentially. One of those is that they need hydrophobic transmembrane domains. So they're going to have hydrophobic parts of the protein. A domain is a specific piece of the protein. We talk about domains in proteins, and they tend to do little different things. So there's a hydrophobic domain, and that spans the membrane. It goes across the membrane. That's the transmembrane part. Some examples of these might be channels that allow molecules to move across the membrane. In this case, we have the hydrogen ions moving through these integral proteins here. Other things might be like receptors. You might have a receptor that takes in a signal on one side and transmits that information into the cell on the other side, or vice versa. And then there are even enzymes that facilitate metabolic reactions. We have things like these transmembrane proteins here in mitochondria that help make ATP. So we have something like ATP synthase. ATP synthase, hydrogen ions flow through it, and ATP synthase is really cool. It actually turns and it catalyzes this reaction, creating ATP from ADP and phosphate here. So that's an integral membrane protein that's bound to the mitochondrial membrane in eukaryotes. We also saw something like the receptors that I talked about in the HIV example. Here's the integral membrane protein, CD4. It's a receptor. It's out here looking for its signal. HIV comes along and it tricks the receptor into letting it in. Here's an epinephrine hormone receptor that takes the signal from outside and it transmits it inside the cell. So when we talk about signaling, right, you get some sort of signal outside your cell and you need to send that message into your cell. Here we have a receptor protein that's an integral membrane protein. Epinephrine binds to the outside and it changes the shape of the protein, causing something to occur inside the cell. Here we have a cascade of things that happen and it goes on to change the cell's response inside of the cell. So this is an example of what we call signal transduction, taking a signal from outside, sending it inside the cell to cause some sort of change. These proteins that are here, like the receptors, they're not always just purely protein. Many of these things also have sugars added to them, so they're glycoproteins, or we have things like lipoproteins and stuff like that. Many viruses and bacteria utilize this trickery to trick our receptor proteins and get inside of our cells. Usually, though, our immune system will catch on to this and start targeting those viral things. But viruses are subject to evolution. A virus would then be rewarded for evolving a new shape of glycoprotein that your immune system doesn't recognize. And it's a constant arms race of your immune system trying to recognize it and it evolving new shapes and continuing to try to do what it does, get inside the cell. If you're interested in viruses, Micro 250 is a great class for that. If you're really interested in healthcare, Micro 111 also, we talk quite a bit about viruses as well. But again, that course is more about getting into one of our many healthcare programs here at CEI. Your membranes need to be fluid, and there are various factors that affect how fluid a membrane is. One of them is temperature. So you're probably aware as things get colder, they get more brittle. That would be real bad news for your membrane, right? If you have a very thick, dense, viscous membrane and it gets cold, it's going to start getting very brittle and it could burst. That is indeed what happens in frostbite. The membrane gets brittle, ice crystals build up, and they actually puncture the membrane, causing cellular death. So you should not go anywhere near frozen water, right? Like, it is very dangerous. Just a few minutes immersion in icy water and you can get frostbite and lose significant portions of your tissue, right? Well, there are organisms that live in the Arctic and Antarctic How do they deal with this? Well, some of it's they have proteins in their circulatory systems that help them not freeze. They're basically anti-freeze proteins. But the other thing that they have is a slightly different membrane construction. So things like cholesterol and unsaturated hydrocarbon tails can make membranes more fluid. So here we can see there's saturated fatty acids here, right? So they're straight and they pack together really well, making this very viscous membrane. If you have unsaturated fatty acids, they have a little kink in them, and that creates space between the phospholipids. That makes the membrane more fluid at lower temperatures. So these organisms have actually evolved phospholipids with slightly different chemistry to allow their membranes to be more fluid at cold temperatures. Okay, before we finish, I want to review here. The head portion of a phospholipid faces towards water because it is A, polar, B, nonpolar, C, planar, or D, isomeric. Go ahead and pause the video, think about that. All right, the answer would be A, polar. Water is a polar substance. The head of a phospholipid is polar, and those two things will interact because of that. All right, so we've covered the structure of the cell membrane now. It is defining the boundary of the cell, right? Everything outside is outside the cell. Everything inside is inside the cell. And it's critical in regulating what comes in and goes out of the cell. The membrane itself, the phospholipids, are a sort of fluid mosaic. They form a bilayer with lots of proteins embedded within them. We have integral proteins that go all the way across the membrane. Those have to have a hydrophobic transmembrane domain. Then we have peripheral proteins that are found on the outside only of the membrane. Phospholipid structure is really important. That amphipathic nature will cause them to self-organize into a lipid bilayer because they're trying to hide their hydrophobic tails. And we talked a bit about how membrane fluidity is determined by the composition of the fatty acid tails. Okay, we're going to talk more about what can and can't go through a membrane and how we get things in and out of the cell in the next few sections.